I want to introduce the first speaker, and Eleanor Jones is from FARA, and she works in the Detection and Surveillance Technologies team. And so she's working on method development for a range of species. And she's going to talk about harmonization of standard laboratory diagnostics in the UK. Over to Ellie.
Okay, well, thank you very much. And yes, good morning, good afternoon, good evening, good night to everyone. I'm presenting on a fairly simple work package, and I'm presenting on behalf of everybody else who's participated in this work package. So as a bit of background, Xylella has fairly distinctive symptoms, but it can't be diagnosed based solely on those symptoms. In order to have a certified positive, we need the result from a molecular method to confirm that we have detection of Xylella. In the UK, any suspect sample would be sent to one of 4 labs. It would be sent to FARA, to Forest Research, to SASA in Scotland, or to AFBI in Northern Ireland. One of the underlying, there are a variety of methods that we could use, a variety of molecular methods that we could use to detect and confirm Xylella. But these different methods are likely to vary in their sensitivity. Even if we were using the same methods, the way that each lab interprets those methods and deploys those methods is also likely to vary. And so potentially the same sample sent to one of those 4, each of those 4 labs has the possibility of producing a different result. So the aim of this task within 2A was to just check how whether the same sample sent to different testing labs was likely to produce a similar result. So the aims of it, it was an excellent opportunity to share expertise, to meet up as the testing labs, and to discuss what we're doing and how we're doing it and the problems that we've encountered, and to discuss which species we've already tested. Further to that, we were then going to compare and collate the molecular diagnostic methods that we're using. Considering those, we could decide whether there was any sort of harmonisation to a standard method was necessary or not. In principle, we could be using different methods, but as long as we get the same results, so a single sample sent in would produce a positive or produce an inconclusive result or produce a negative, then that's the main thing. So we wanted to have an interlaboratory test process where we sent the same sample to different labs to compare the results that people were getting. So the first exercise was the data sharing. So we had a series of meetings in person, which was very nice and seems a bit unlikely now. And we sent around our standard operating procedures so that other people so that each lab could see what everyone else was doing and the actual breakdown of those steps. And we collated lists of the methods that people were using and lists of the species that people had tested. It's probably worth noting that when we say this, we're talking about none of us have got samples that actually have Xylella. We have leaf tissue, for example, from each of these species, and then we spike on Xylella and see if we can detect it. So for people who aren't molecular biologists, the workflow is that you receive a sample, so you get some leaf material or possibly a plant that's been sent in. You then take some of that sample, you have to extract the DNA from it, and then you have to try and detect whether you've got Xylella within that sample. So that's the breakdown of those steps. If we compare the sampling, FARA is using a standardised 0.6 gram sample. SASA and FR are using between 0.5 and 1 gram, depending on the leaf, depending on the plant material. There's a slight difference in what people are likely to be testing. So FARA is more likely to be testing lavender than Forest Research. Forest Research are likely to be testing trees. SASA are going to be testing whatever is sent to them. The DNA extraction step is the one where we diverge most on methods. So FARA and SASA are both using CTAB methods. All of us are homogenizing the leaf tissue. It's a very necessary step. And I've put an asterisk next to the CTAB because although we're both using CTAB methods, we are using different variations of CTAB, and FR are using QuickPick. In terms of the detection, we are all using the same frontline tool, which is real-time PCR, and we're all using the same assay. We're all using Harper et al., which is good. If we had a first intersection, or if we had an important sample, we'd have to do confirmatory testing on it. And we're all using the same sorts of second line testing, so conventional PCR. But we were all in agreement that if we had that first intersection or that very important sample, we would probably use every single tool we have to examine that sample as much as possible. If you think about interlaboratory testing, there's a variety of ways you can do it. The simplest, I've just shown as an orange bar at the bottom, called it Interlab Test 1, is that we send around DNA and test that everybody is getting similar results from their real-time PCR assays. A more comprehensive test, which I've labelled here as Interlab Test 2, would be to send out leaf material that's got some sort of Xylella on it. And then you're testing both, you're testing the extraction step and the detection step. So we did the first interlaboratory test. Ferrer prepared Xylella fastidiosa DNA and lavender DNA in quite large volumes in fixed concentrations. These were then sent out to...
We are waiting for a sample that will come to her home.
Sorry? Right. So we circulated the test material to FR SATA, and we also sent it to AFB in Northern Ireland because they were another sampling laboratory. Ferrer did a mock posting, so we stuck it on a desk and we waited until everyone else had received their packages, and then we put it back into the freezer. And in that way, we're trying to control that everybody has material in the same condition. All the laboratories did 3 dilution series. So we sent the instructions so that everyone was doing exactly the same dilution series. And we did a dilution series of Xylella fastidiosa in water. So we diluted it down till we couldn't detect it anymore. We did a dilution series of lavender DNA, where that was diluted down. Everybody is using a plant control assay, so that when we say we're testing for Xylella, we're actually testing 2 things. We test that we have got whether we've got Xylella, and we're also testing whether we've detected plant DNA. And that way, if we get a negative on our plant test, we know that the assay has failed. So it's just effectively a control. The final dilution series that everyone did was to dilute Xylella fastidiosa DNA against a background of lavender DNA, and that more closely mimics the actual samples that people will be testing. We're almost never looking for Xylella in a dilution of water. We're often looking for Xylella in the background of plant DNA. The interlaboratory test, if you can't see the detail of this, but that's fine. I've highlighted in orange the point at which detection drops out. So where people are no longer detecting Xylella fastidiosa. It's indicated as, the results are shown as cycling thresholds. Sorry, the results are shown as the dilution series going down, and you can see that we're all broadly getting the same sensitivity, that the assay is dropping out more or less at the same point in the dilution series. In green, I've shown where people are calling the last positive, and everybody is using a threshold, so a cutoff, to say if the result is below that number of PCR cycles, then we've got a positive. If it's above, but we're still detecting something, then it's a retest or it's an inconclusive result. And one thing that came out from this was that the different labs were using different thresholds. So although we had the same sensitivity, we were actually calling the results slightly differently. That was very simple. We agreed to harmonise on cutoff and the way that we call those results. One other thing that came out from this was that Forest Research were multiplexing in their plant control, which meant that they were just doing a single assay, and we thought we would take that one forward and other labs would try the same. Right, my final slide. I think I'm doing okay for time. So we were going to do a second interlaboratory test where we were going to send out spiked leaf tissue, but coronavirus happened. and everybody's labs were shut down. And when they reopened, or they were stripped down to the minimum, and when they reopened, we were all just trying to do our statutory work. So we haven't managed to progress this one. We thought we'd got quite far, but then we lost some of the plants from the greenhouse. And so we have to wait for those. The intention in this test is that we will take probably nerium leaf tissue, a standardized amount of leaf tissue. We will spike on a fixed volume and concentration of Xylella fastidiosa culture, and then put it into a grinding bag or a grinding tube and send those out to the individual's labs. So each lab will then do the DNA extraction step followed by the detection step. One thing to note, we will definitely make sure that the Xylella is dead before we do this part of the exercise. But yes, sadly, we haven't managed to proceed with that step, but hopefully in the new year, we'll be able to progress this one. And that is essentially what we've been up to in work package 2A. My acknowledgements. It's been a lot of work from all the different partner labs, and thank you to the JIC for doing all the organisation and the IT, and to all the funders. Thank you. And I can stop sharing my screen. Any questions?
We have time for a couple of questions.
Okay.
If no one comes, I'll ask a question. Do you think that the DNA extraction method can have an effect on then the end result? Or do you think all the methods that are being used might be quite equivalent?
It is likely that they'll be different, but it's not necessarily entirely predictable how they'll be different. Also, we're likely to be testing different materials. So the tree materials can be very different in character to some of the plants, to the softer plants that Farah will be dealing with. So in some ways, we can test the results and we can see that we're returning the same results, but that might also vary depending on what we're testing. And in this case, we're testing lavender or nerium, which Farah's methods are optimized for.
We have a question here from Dominique Hare. Can you comment on putting subsamples from multiple plants into the same sample?
I am not the expert, I'm afraid. I'm just the person talking.
Have you tried that or not?
I am certain that within Farah we have tried this. Yes, so I think we take subsamples from a single sample. We all take multiple subsamples and pull those. I don't know if that's the talk. That's the question. Jennifer Cole and Adam Brining are our technical experts who deal with most of our samples.